Hi, everybody. My name is Veronique, and I am the Scientific Application Manager at BPS Bioscience. In this poster, I am presenting some of our BCMA-focused research tools that BPS scientists have generated to allow investigators to focus on the critical questions. First, I will describe our VCMA-based assays. They quantify the interaction between BCMA and ligands BAFF or APRIL. The principle of these assays is simple. One partner is coated onto the plate. The other partner, purified and labeled with biotin, is added to it. After washing, structure-activating HRP is added, then the HRP substrate. The chemoluminescence signal is directly proportional to how much binding has taken place. Example shown here and here. So these assays are particularly useful to screen for inhibitors of the interaction between BCMA and these ligands, to validate blocking antibodies, or to titrate antibodies and inhibitors, as illustrated in these two examples. BPS biologists have also generated a series of BCMA-expressing cells listed in this table. The BCMA CHO cells were selected for high, medium, or low expression of BCMA compared to the parental cells and shown here in flow cytometry. Other cell lines also express luciferase in addition to BCMA. On the right, I am showing an example of the human B cells RPMI 8226, which constitutively express BCMA and represent a physiologically relevant model. They were stably transfected with luciferase. Now, since the luciferase activity is directly proportional to the number of cells, as shown in this graph, the cells are ideal for co-culture experiments. For example, when testing the cytotoxicity effect of anti-BCMA immune cells. The experiments shown on the lower portion of the panel demonstrate how Birayovich et al. Used our BCMA CHO cells to validate a new monoclonal antibody for CAR T-cell development. This study was published in Cancers. Several antibodies were tested against the BCMA CHO cells using flow cytometry shown here. Then the authors measured interferon production by the CAR T cells co-cultured with CHO cells or with CHO BCMA cells. Then they measured CAR T-cell induced cytotoxicity in co-culture experiments. As shown here, the CAR T cells that express the right antibody construct killed the BCMA CHO cells, but they didn't kill the control CHO cells. Other tools that we offer include biospecific and trispecific T-cell engagers. These are composites. They're constructed from antibodies that recognize distant antigens. For example, one domain of the biospecific antibody recognizes antigen CD3 on the surface of T cells, whereas the other domain recognizes BCMA present on tumor cells. So the bispecific antibody physically brings the cells in close proximity. This results in the activation of the T cell, which triggers cytokine release and cytotoxicity. In other words, it kills the tumor cell. Our trispecific antibody is designed to bind CD3 on T cells and both BCMA and CD19 on the tumor cells. This graph, for example, illustrates an experiment in which CD19 overexpressing CHO cells were co-cultured with CD3 positive Jurkat reporter cells, and then the trispecific antibody was added. Now, in these T cells, the reporter activity correlates with T cell activation. So what we conclude here is that the T cells were activated in the presence of the CD19 CHO cells by the trispecific antibody, while they were not activated in the presence of the control CHO cells. Finally, I will quickly go over some of our TCR-based tools. BPS offers a range of TCR-based products, including lentiviruses for scientists to make their own TCR activator or TCR knockout cells. The TCR knockout cells use the TCR CRISPR-Cas9 lentivirus, either in the integrating or the non-integrating version. The TCR activator cells, like the TCR CHO cells, stably express TCR activator molecules on their surface, and they activate T cells through binding with a TCR. They can be used as positive controls or in optimization experiments. On the other hand, the TCR knockout Joker T cells have lost the expression of the main alpha and beta subunits of TCR. This was confirmed by flow cytometry. So in this experiment, the parental cells are shown in purple and they do express TCR, whereas the knockout cells are shown in green and have lost TCR expression. These cells can be used as control, for example, when generating CAR T cells, or they can be used to determine specific T cell cytotoxicity in co-culture experiments. To conclude, BPS supports scientists at all phases of their research projects to accelerate the discovery and the development of new immunotherapies targeting a growing list of potential targets such as BCMA or CD19. We offer antibodies, biospecific and trispecific T-cell engagers, cell lines with or without reporter genes, biochemical assays, TCR-based tools, lentiviruses, and customized services. Thank you for listening.